The presentation by Sergei Petoukhov, Elena Petukhova, V.Svirin, Vladimir Verevkin about algebraic harmony in genomes of higher and lower organisms. The creators of quantum mechanics, Jordan and Schrödinger, pointed out the main difference between living bodies and inanimate ones. In living bodies, genetic molecules play a dictatorial role. Inanimate objects have no such dictatorial molecules, since they are governed by the average random motion of millions of particles. The study and use of the properties of DNA molecules with their nucleotide sequences, which carry genetic information, are some of the key tasks of modern science. See the history of quantum biology in this article. The report presents our research on informational features and dictatorial influence of DNA heredity molecules on living organisms. They talk about universal hyperbolic rules and algebraic harmony in the informational organization of genomes of higher and lower organisms. Hereditary information is recorded in DNA molecules in the form of long sequences of 4 nucleotides, molecular letters, adenine A, thymine T, cytosine C, and guanine A. G. These DNA sequences for each genome of many organisms are presented in the open access in the gene bank in a standard form, which gives the impression of a chaotic DNA text of 4 letters with a length in thousands of pages. But as known, these genomic sequences of 4 letters are not at all random. They contain many complementary palindromes, etc. Let us now turn to the universal hyperbolic rules that we have discovered for genomes of higher and lower organisms. Let's start with an example of the DNA of human chromosome number 1. Whose chain contains about 230 million nucleotides, A, T, C, and G. Let's first calculate the number of nucleotides A. Then, in the chain of its doublets, calculate the number of all 4 doublets starting with A, that is AA, AT, AC, AG. Then, in the chain of its triplets, calculate the number of all 16 triplets, starting with A, AAA, AAT, etc. And so on for each of the set of 4 in the power n minus 1 plets or oligomers, starting with A. Take here n is equal 1, 2, 3 till 20. We get the following 20 numbers. It is striking that all 20 points with these coordinates practically coincide with the hyperbolic sequence shown here, which numerator is the number of nucleotides A in this DNA. The deviations of real quantities from this sequence are hundreds of a percent and lie in the range minus 0.030% till 0.024%. Knowing the number of nucleotides A in the genomic DNA text, it is possible to predict with high accuracy the values of 19 other named sums of n-plets, oligomers of length n. Since, as it turns out, there is a cooperative connection between them. This new author's method of analyzing and comparing DNA text is called the method of oligomer sums. Similar results are obtained in the same chromosome when calculating the total number of N-plates, oligomers, which start with any of the other three nucleotides, T, C, or G. The resulting four hyperbolic sequences for the human chromosome number 1 differ practically only in the value of their numerators, which are equal to the amounts of corresponding nucleotides. The human genome contains 22 autosomes and 2 sex chromosomes X and Y. They all differ from each other in length, kinds and numbers of genes in them, molecular weights, etc. The analysis of all 24 human chromosomes by the oligomer sums method showed that in all of them, similar hyperbolic sequences are realized with high accuracy, with individual values of the numerators for each chromosome. Similar hyperbolic sequences of oligomer sums were obtained in all chromosomes in all tested other eukaryotic traditional for genetic research. Drosophila having 7 chromosomes, mouse 21 chromosomes, nematoda 6 chromosomes, plant Arabidopsis thailandana 5 chromosomes. We carried out also, by the method of oligomer sums, analysis of all 19 genomes from different groups of bacteria and archaea listed in the article. All the genomes of prokaryotes demonstrated with high accuracy the same phenomenon of the interrelation of oligomer sums in the form of their hyperbolic sequences. We were especially interested in the genomes of organisms capable of living in extreme conditions, such as powerful radiation, high and low temperatures, acidic and alkaline environments, in the vent of volcanoes, etc., including the genome of the microorganisms surviving in conditions of radiations thousands of times higher than the level fatal to humans. Our selective study of the genomes of many such extremophiles showed that all of them are also subject to general hyperbolic rules of the interrelation of oligomer sum. Until now, not a single genome of eukaryotes and prokaryotes has been found in which oligomer sums would not be linked with high accuracy, about 1% or less, by the named hyperbolic rules. It should be emphasized that the genomic hyperbolic rules are cardinally different from well-known hyperbolic Zipf's law. In linguistics and other fields, Zipf's law speaks of the frequency of encounter of separate words or other separate objects. In contrast, the genomic hyperbolic rules of oligomer sums focus on sequences of the total amounts of special N-plates, oligomers, that is, on the relative number of not separate oligomers, but the whole sums of sets of different N-plates distributed inside the genomic sequence, where each separate nucleotide is a part of many oligomers set existing simultaneously. Each nucleotide is a distributed participant of many members of the appropriate genomic oligomer sum sequence at once and makes a contribution to each of them. Genomes and the harmonic progression, next section. The model hyperbolic sequences serve as mathematical standards for general phenomenological rules of oligomer sums in eukaryotic and prokaryotic genomes. These hyperbolic sequences differ from each other only in the magnitude of numerators in their expressions. By appropriate scaling of numerators, each of these genomic hyperbolic sequences is transformed into the historically famous harmonic progression. The name, the harmonic progression, comes from its connection with the series of harmonics in music, or lengths of standing waves in a stretched vibrating string. The harmonic progression was studied by Pythagoras, Leibniz, Newton, Euler, Fourier, Dirichlet, Riemann in relation to different mathematical and physical problems. Now, the harmonic progression shows itself unexpectedly in all tested genomes. The harmonic progression has interesting mathematical features, which can be useful for developing new classes of models of genetically inherited physiological structures. For example, its term is the harmonic mean of the neighboring terms. Knowing any two adjacent terms of the harmonic progression, one can recurrently construct all the harmonic progression using the expressions for the harmonic mean. The sums of the first members of the harmonic progression are called harmonic numbers. The generalization of the harmonic series is known as the Riemann zeta function. The series of harmonic numbers serve as the discrete analog of the continuous function of natural logarithm. This in particular connects the harmonic progression with the logarithmic Weber Fechner law, which is the main psychophysical law and dictates information features for all inherited sensory channels, vision, hearing, smell, etc., whose organs, eyes, ears, nose, etc., very differ each other in appearance. It testifies that genetic and different psychophysical levels of inherited biological informatics are structurally intercorrelated on the algebra-harmonic basis. The harmonic progression and the series of natural numbers, 1 through 2, 3, etc., are projectively identical and are transformed each into other by projective transformations, since in each of them the cross ratio, which is the main invariant of projective geometry, for any 4 adjacent members is equal to 4/3. This gives an outlet on the rich topic of non-Euclidean biosymmetries in genetically inherited biological structures, including in particular a known projective nonlinear changing of proportions of the kinematic scheme of human body in ontogenesis. The presented data on the universal hyperbolic rules of the oligomer harmonic organization of eukaryotic and prokaryotic genomes say that these global genomic algebraic invariants remain unchanged over millions of years of biological evolution, when millions of species of living organisms die and new ones appear, although locally genomic sequences are modified under the action of mutations, the pressing of natural selections, etc. The totality of the results obtained in our laboratory indicates that the genomes of all analyzed organisms appear to be a regular algebraic fractal-like network with the important participation of the harmonic progression in the interconnections of parts of the network at different levels. That allows us to talk about the algebraic harmony of living organisms. Some important results were received from the quantum information model of long DNA sequences, which allows non-trivial predictions. See this article. Thus, algebraic and quantum biology is developed further and further. At the tip of the pen, it is now possible to predict with high accuracy non-trivial quantitative characteristics of genomic sequences. Additional quantitative data for our presentation can be found in the following articles. Conclusions. Firstly, the presented results give pieces of evidence on the existence of universal hyperbolic rules in the oligomer harmonic organization of eukaryotic and prokaryotic genomes. Secondly, the results allow us to talk about the algebraic harmony of living organisms. Thirdly, the results are useful for further development of algebraic and quantum biology. Thanks for your attention.